So now that we've reviewed the general characteristics of eukaryotic cells, let's take a look into some of the specific cell types that we'll be studying in microbiology. The kingdom fungi is where we should begin. Now, fungi were originally classified with green plants, along with algae and other bacteria. This is a long time ago. And then they were split off and put in the category of protists, along with the algae. But we realized that there were so many unique qualities of fungi. They were distinct from animals, they were distinct from plants, and they didn't really fit into the protist group. So they must have their own kingdom. This kingdom is known as the kingdom Mycetae, also known as fungi. It's a very large kingdom with a great variety and complexity of organismal types. There are approximately 100,000 species. A lot of them are macroscopic, like the mushrooms that you might put into your pasta. However, many are microscopic, like yeasts that you use to generate leavening in bread. The majority of these microscopic fungi are unicellular or colonial. There are several complex forms, like the mushrooms and the puffballs of the macroscopic fungi, that are considered to be multicellular organisms. Let's take a closer look at the microscopic. There are two forms. There's the yeasts and the molds, or hyphae. Yeasts are single-celled organisms. They're round to oval in shape. You can see a generic yeast shown right here. It's a single cell, and they're round and oval to shape, and they usually reproduce with asexual reproduction. That asexual reproduction involves formation of a bud that later breaks away from the cell. The other type are molds, or hyphae. The hyphae are long thread-like cells, and they're found in colonies. You can see one of the colonies of these thread-like cells here, magnified here. And then you can see a diagrammed version of these hyphae down here. Now, some yeast cells actually form chains, as you can see here, by buds that remain attached in a row and never actually separate. These are called pseudohyphae. Now a little bit about fungal nutrition. First of all, they're heterotrophic, much like ourselves, which means they obtain their nutrients from a variety of different substrates or materials. They're non-photosynthetic, so they certainly don't belong with algae or plants. And they're saprophytic. That is, they are saprobes, which means they obtain their substrates or their nutrients from remnants of dead plants and animals. They're decomposers. Some are parasites. When a fungus causes an infection in an animal or plant, as in a parasite, that infection is called a mycosis. Here are a couple of examples of parasitic fungi. You can see in this image here the rhizopus fungi that's growing on these raspberries. The fungus will penetrate the substrate and secrete enzymes that form much smaller molecules that can then be absorbed by the fungal cells. Fungi have many different enzymes that can digest a huge number of substances. For example, they can digest feathers and hair and skin, wood, even rubber. In the image of this foot here, which is kind of gross, you can see a soil fungus has embedded itself onto the substrate or the skin of the foot and has begun to digest away portions of the skin to keep itself alive. This is an example of how it could be a mycosis, a fungal infection. Now let's take a little closer look at the organization of some of our microscopic fungi. Most of them grow in loose associations or even colonies, as you can see over here. Yeasts live in colonies that are much like the colonies of bacteria, so they have a soft, uniform texture and appearance. However, the hyphae, or filamentous fungal colonies that we commonly know as mold, have a more cottony, hairy, or velvety texture. When we look at these hyphae, we'll see that they come in two different forms. We have septate hyphae over here on the left, and then non-septate hyphae on the right. The septate hyphae are cells that are divided into segments by septa. They're like cross walls that divide the cytoplasm into segments. They usually aren't complete divisions, so the cytoplasm can still flow between them, but they're enough to keep the major cellular components from wandering around too much throughout the cellular structure. The non-septate hyphae are essentially one long continuous cell, and the components can move around more freely throughout the whole entire cell. You'll notice that there's a woven intertwining mass of hyphae. These make up the body or the colony of the mold and are called a mycelium. Another way of thinking about hyphae is whether they're vegetative or reproductive. The vegetative hyphae or mycelia are the visible mass or growth on the substrate surface. These are the cells that penetrate the substrate and digest and absorb its nutrients. The reproductive or fertile hyphae form from the vegetative hyphae and are responsible for the production of spores. In this figure, you can see the vegetative hyphae here with some submerged in the substrate. Over on the other side, we can see these reproductive hyphae, which are producing spores. So in order for many fungi to reproduce, they have to either fragment or produce spores. So most can propagate by growth of the hyphae or fragmentation. And in fragmentation, we have a separated portion of the mycelium, which can actually generate a whole new colony. However, the primary reproductive mode is through the production of spores. Now, fungal spores are not to be confused with the non-reproductive spores that we saw in bacteria. They have several functions. One is multiplication of the organism, which of course contributes to survival. And then we have genetic variation through the production of spores, especially the sexual spores, as well as dissemination or spreading around of the fungus to new environments. There's a large diversity in the spores among fungi, but we can look at a general grouping of spores, first asexual and then sexual. Here are some asexual spores. They're known as sporangiospores. They're formed by successive cleavages within the sporangium. Here you can see the production of sporangiospores within the sporangium. So basically, we have cell division and cell division inside the sporangium. The sporangium's attached to a sporangiophore, which is essentially the stalk of the sporangium. When the sporangium ruptures, all these spores, or sporangiospores, are released into the environment, and thus we have asexual propagation of the fungus. The other means is by conidiospores, or conidia. These are free spores that are developed either by pinching off of the tip of fertile hyphae or by segmentation of a vegetative hyphae. There are many different forms of conidiospores. You can see some examples over here on the right. Finally, let's take a look at sexual spores. Fungi can propagate themselves so easily with asexual spores. So why is it that we would even have sexual spores? The key here comes in acquiring genetic variation. When we link the genes from two parents, we create offspring with a combination of those genes. And this combination could be potentially advantageous and thus allow for better survival of that particular species. The majority of fungi produce sexual spores at some point in their life. Now, this can involve simple fusion or fertile hyphae of two different strains, or it could be as complex as the union of differentiated male and female structures that lead to the development of fruiting structures. Now, mushrooms, for example, are the fruiting structures of some macroscopic fungi. The whole organism lives below the surface of the earth. And when it's time for sexual reproduction, it pops up mushrooms. When we consider medical specimens and identifying and cultivating them to establish the cause of some condition, we'll find that more often than not, the microscope still remains the primary means of identification. We'll use special types of media and observe the specimens macroscopically and microscopically. As you can see from these figures below, fungi have a very distinct and prolific appearance under the microscope. There's other reasons for this, though. Usually we use the sexual spore-forming structures and the spore types to classify fungi into general groups. However, the sexual spores are rarely detected in the medical lab setting, so we have to use asexual spore-forming structures, as well as hyphal types and colony texture and pigmentation, as well as some of the physiological characteristics, and on occasion the genetic makeup, to establish the type of fungi that are in a particular specimen. In contrast, when we're looking at bacterial and viral identifications, we rely much more increasingly on molecular techniques. But fungi have such unique and diverse makeups that we can see them clearly under the microscope. Nearly all fungi are free-living and don't need a host. However, some of them are parasitic. Human infection by pathogenic fungi usually occurs through accidental contact. Generally, we're pretty resistant to these fungal infections, except for two main types. There are primary pathogens, which can sicken even a healthy person. And then there are opportunistic pathogens, which attack people who are already weakened in some way. So far, there are only about 270 species of fungi that have been found to be able to cause human disease. The list of opportunistic fungal pathogens, however, has been increasing due to all the new medical techniques that keep immunocompromised patients alive longer. Even harmless varieties of fungi can cause opportunistic infections with patients who have been weakened by AIDS, cancer, and diabetes. Mycoses, or fungal infections, vary extremely in the way that the agent enters the body and the degree of tissue involvement. You can see a few examples of the major fungal infections in humans in this table below. Now, fungi are not only responsible for infectious medical conditions. There are other conditions, such as toxicity and allergies that are caused by fungus. Fungal cell walls give off chemical substances that can cause allergies, and some toxins that are produced by poisonous mushrooms can cause neurological disturbances or even death. Hallucinogenic mushrooms fit into this category. Fungi also have a substantial role in agricultural damage. Pathogenicity in plants, such as corn, or even the animals that eat the corn, and in fruit, causes economic hindrance in agricultural industry. For example, about 40% of the yearly fruit crop is consumed by fungi during its storage and transport, rather than the humans that it was grown to feed. Now, on the brighter side, fungi do have some benefits. They're heavily involved in decomposing organic matter, which returns the essential minerals and nutrients back into the soil so that they can be used by future growth. They also form stable associations or symbioses with plant roots. Mycorrhizae are these associations, and they increase the ability of plant roots to absorb water and nutrients, so they're integral in our primary production. Recently, there's also been a surge in biochemical potential of fungi to produce large quantities of things like antibiotics and alcohol, organic acids, as well as vitamins, through some of the new innovations in biochemical techniques. In addition, some fungi are even eaten or used to flavor foods. For example, many cheeses and soy sauces and cured meats derive their unique flavors from the actions of fungi. One that we're particularly interested in is Saccharomyces. This is the yeast that's producing alcohol in beer and wine, and also which causes bread to rise. So there's an example of where fungi are integral in our society. Before we move on, let's have a quick concept check. Which would not be found in a fungal cell? A cell wall, B, nucleus, C, chloroplasts, or D, an 80S ribosome? Well, we know that fungi have cell walls, so we can cross that one off the list. We also know that they're eukaryotic, and thus they have a nucleus. Also, because they're eukaryotic, they have the 80S ribosomes, as opposed to the prokaryotic 70S ribosomes, so we can take that off the list. So that leaves us with chloroplasts. This makes sense because we've already learned that fungi are non-photosynthetic.